Structure and functions of immune system. The lymphoreticular system is responsible for immune response, and it consists of the lymphoid and reticuloendothelial components. The lymphoid cells, lymphocytes, and plasma cells are responsible for the specific immunity. The phagocytic cells, polymorphonuclear leukocytes, and macrophages, which are part of reticuloendothelial system, are mainly concerned with this scavenger functions of elimination of microorganisms and other foreign particles from blood and tissues, thus contributing to non-specific immunity. The lymphoid system, lymphoid cells, example lymphocytes, plasma cells. Lymphoid organs, central lymphoid organs, example thymus, bursa of fabricus in birds, bone marrow in mammals. Peripheral secondary lymphoid organs, example spleen, lymph nodes, mucosa associated lymphoid tissue, lymphoid tissue in guts, lungs, liver, bone marrow. Types of immune system. The specific immune response to an antigen can be of two types: the humoral or antibody mediated immunity. It is mediated by the antibodies which are produced by the plasma cells. The cell mediated immunity, it is recognized by the sensitized lymphocytes. The central lymphoid organs are structures in which the precursors lymphocytes proliferate, develop, and becomes immunologically competent. After acquiring immunological compatibility, the lymphocytes migrates to peripheral lymphoid organs where appropriate immune responses occurs when exposed to an oxygen. Cells of lymphoreticular systems. These consist of structural cells, reticulum cells, endothelial cells, and fibroblast, and immunologically competent cells, lymphocytes, plasma cells, and macrophages. In the circulating blood, about 70% lymphocytes and T lymphocytes, 20% B lymphocytes, and 10% of null cells. Difference between the T and B cells. The characteristic feature of to differentiate T cells and B cells are shown in the table. T lymphocytes. The T lymphocytes can be classified into different subpopulations or subsets based on functional capacity and on surface and antigenic determinant. On the basis of functions, various subsets are as follows. Regulatory T cells, effector T cells, regulatory T cells, T helper cells, CD4 cells, T suppressor cells, effector T cells, cytotoxic T cells, delayed type hypersensitivity cells, mixed lymphocyte reactivity. On the basis of surface antigenic determinant, the surface antigen on the T cells can be detected by monoclonal antibody. These thymic antigens or markers were previously designed by the letter T1 to T11. The markers are now designed as CD, the subsets of the cells in man as shown in the figure. Null cells, the small portions of the T lymphocytes which are neither T cells or nor B cells are known as the null cells. Among the null cells, the killer cells, K cells, natural killer cells, and the lymphokines activated killer cells, LIK cells are recognized. The K cells are now classified with the NK cells. Killer cells, K cells. These lymphocytes possess surface receptors for Fc portion of IgG. These cells are capable of killing target cells sensitized with the IgG antibody. They do not require complement for such lysis or killing. These are responsible for antibody-dependent cell-mediated cytotoxicity. Natural killer cells. The natural killer cells are cytotoxic for virally transformed target cells, certain tumor lines, and are involved in allograft rejections. The natural killer cells, NK cells, are found in spleen and peripheral blood. The action of natural killer cells is independent of antibody in contrast to killer cells which are dependent on antibody. Their activity is natural or non-immune as it does not require sensitization by prior antigenic contact. They release several cytotoxic factors. The natural killer cells activity is enhanced by interferons. It is believed that NK cells plays an important role in antiviral antiviral and anti-tumor immunity. The lymphokin activated killer cells, these are NK cells treated with interleukin 2, which are cytotoxic to tumor cells without affecting normal cells. B lymphocytes and plasma cells. The antigenically simulated B lymphocytes undergoes blast transformation to become plasma cells. The plasma cells can synthesize an antibody of a single capacity of a single immunoglobulin cells, IgM or IgG or IgA. The interaction of T lymphocytes and B lymphocytes in cell mediated immunity, anti-humoral or antibody mediated immunity is shown in the figure. Major histocompatibility complex, the transplant from one individual to another member of same species allografts are recognized as a foreign and rejected. The antigen responsible for allograft rejection in inbred mice were identified and led to the discovery of major histocompatibility complex. The MHC in humans is known as the human leukocyte antigen complex. The major transplantation antigen of man includes carbohydrate antigen of erythrocyte blood groups and glycoprotein antigens of cell membrane HLA system. HLA complex, the histocompatibility antigen means cell surface antigen that evoke immune response to an incompatibility host resulting in allograft rejection. These alloantigens are presented on the surface of leukocyte in man and are called human leukocyte antigen HLA, and the set of gene coded for them is named as HLA complex. The HLA complex of a gene is located on a short arm of chromosome 6 and it is grouped in 3 classes. Class 1 HLA-A, HLA-B, HLA-C. Class 2 HLA-DR, HLA-DQ, HLA-DP. Class 3 the complement loci encoded for C2, C4 and factor B of a complement system and tumor necrosis factor alpha and beta.